Welcome to this lecture on adaptation of animals. Now we will continue on the reveal the mysteries of the muscles and now to compare the fast and slow skeletal muscles. We have dealt this difference already when we were discussing on the how to increase the metabolic rate when we are running as fast as possible. And we learned that, okay, if we are running very fast, we are using glycolytic pathways, and then we can't run for ages because we have lactate, etc. And then we were dealing this same thing already when we were discussing on the tunas that have a higher temperature in some areas because they have oxidative muscles there to produce heat. But actually, the difference between the fast and slow muscles is much more difficult. So in the ancient time, it was very clear, okay, all the skeletal muscles are either fast or slow, but actually they are like a mosaic. So each skeletal muscle of a mammalian looks like this. So if it's red, it means that there is a lot of iron, meaning that there is a lot of myoglobin. If there are black dots, there are a lot of mitochondria. So with this kind of staining, you can see that some muscle cells are pale, almost yellow. They're on the left-hand side. So these fast glycolytic fibers, they are large cells. There's only few mitochondria. And then there in the bottom, there are some red cells, smaller, and a lot of these black dots, so a lot of mitochondria. So these are these slow oxidative fibers. And then on the right hand side there are some orange, so there are some myoglobin, but also some black dots, so the mitochondria. So we have some kind of combination of these two on the same cell. So these are fast oxidative glycolytic cells. And so we have at least 3 different types of skeletal muscle cells. One is slow and 2 are fast. Okay, the difference between fast and slow is perhaps related on the speed to make the contraction. And that's then related on the speed of the myosin ATP use. So the ATPase activity in these slow oxidative cells is slow. So they are using less ATP per second, meaning that the myosin and actin are moving slower on each other, and then the contraction happens slower, and then the speed to reach the peak tension is slower. But that also affects that, okay, when you have the peak tension, then the release is also slower. So that's why the whole twitch is longer in these slow oxidative cells. And part of that is related that not only that the tension happens slower, but also the calcium uptake is slower. So when you are releasing calcium from the sarcoplasmic reticulum, then you have to pump the calcium back to the star. And if this process is slower, then this twitch, the whole contraction is slower. But actually this slow process in these slow oxidative cells is a benefit. So they can resist all the fatigue. So meaning that they are not getting tired. They are not losing power if they are used for a long time. So that's why you can use these slow oxidative cells almost continuously all this day. But these fast glycolytic cells that are very fast to contract, but very fast to relax, but they lose the power, the force, quite fast. And not only we can compare that, okay, these are slow and these are fast, but the typical way to compare them or to divide is that, okay, we have red cells and white cells. But nowadays, the division is used, they use these kind of codes. So the type 1 in the literature, it's mean that, okay, that's the slow oxidative cell. Then we have type 2A, type 2B. They are the fast oxidative glycolytic and fast glycolytic cells. But between them, there might be type 2X or even type 2D. So there might be 4 or 5 different types of skeletal muscles. And most probably, these type 2, they are a continuum from quite oxidative cells to almost completely glycolytic cells. So even then, the fast oxidative glycolytic cells, there can be, the tension can be intermediate or fast, or something between them. So it's much more complex so you can't just say that, okay, this is the slow and this is the fast, but that we have at least 4 or 5 different types. And okay, if it's oxidative and glycolytic, it most probably it's something to deal with the metabolism. So these oxidative cells have a lot of mitochondria. Because there is a lot of mitochondria, there is also need for oxygen store, a lot of myoglobin. And because they are relied on the oxygen, there's also more capillaries around them. And the cell size is smaller. So the diameter is smaller, so because the oxygen from the capillary to the center of the skeletal cell goes by diffusion. And if you have a large cell, it takes too much time to get the oxygen in the center. And of course, if they are oxidative or glycolytic, there are differences in these enzymes. So the glycolytic enzymes, they have much more these glycolytic enzymes in these glycolytic cells. So then they can even upregulate the glycolytics, so to produce more ATP during the exercise, but only for a short period. But then in comparison, then if you have a lot of mitochondria, you have oxygen store, etc., you are able to produce a lot of ATP with this oxidative phosphorylation. And one interesting thing is that, okay, these slow oxidative cells, although the ATP production with the oxidative phosphorylation is the best way. You can produce a lot of ATP, but still the force per cross-section area is quite low. So they are not very efficient muscles. They can work for a long time, but they can't weight large weights. So you can't lift large weight with this. And so of course the cross-section area is important, because if you have very wide whole muscle, then you can produce more force. But that's why usually the force is divided by the cross-section, so you can compare similar size cells, similar size muscles to each other. So these oxidative cells have low force, they are not very strong, but these fast glycolytic cells, they are very strong. And that's why the functions are different. These slow oxidative cells are used for to stay in one position. So these cells are just contracting a little bit to form the tension, but not for moving. So they are keeping us in the same position. But then these fast oxidative glycolytic cells we use for standing, so contracting a little bit more to stretch the ends of the muscle, and they are used for walking, but also for rapid repetitive movements like driving bicycle. And then we have these glycolytic cells that we are using for real exercises, so running as fast as possible, etc. But of course we can do that only for a short period, so that's why the frequency of use by animals is very high in the slow oxidative cells. We can use them all day through, and these fast glycolytic cells only for a short period. Okay, so there are several differences between different types of cells. but on what is the basis of that? One thing is that, okay, there's a difference in the ATP consumption in the myosin. And if we look at, okay, what kind of myosin we have in the mammalian body, we have several different genes that are coding myosin. So that's why we have several different isoforms of myosin in our body. And for example, there are different isoforms in type 2A, type 2X, and type 2B muscle cells. And these isoforms have different ATP activity. So they are using ATP, more or less ATP per second. And if you compare, there in the one, that means the slow one, the slow skeletal muscle. The ATP consumption is about 1/4 of these type 2 cells. And even among the type 2, there is a huge difference on the ATP consumption. And this affects then that there are differences in the shortening velocity, how fast the muscle is contracting. So these type 1 cells are contracting slower. And the filaments are contracting slower. But as you can see over here, there is a mouse, rat, rabbit, dog, and human. There's species-dependent variation also. For example, in the difference in the shortening velocity, there in the middle. Type 2A in the human, on the right-hand side, it's quite low versus in the mouse. And it's actually at the same rate as in the type 1 muscle cells in the mouse. So that's why some animals are able to run faster, to move faster. And that's why we humans, we are not the fastest animal in the world. Okay, myosin is different in fast and slow muscle cells. Okay, what about the other elements of this contraction machine? Well, also the myosin light chain and the heavy chain, they have fast over here in red and slow over here in green isoforms. So not only the heavy chain, but also the light chain. And also the supportive protein titin is different in slow muscle and the fast muscle. Although we don't know exactly what is the purpose of the titin. But then also in the thin filament, there are differences. Well, the actin is the same, but tropomyosin and troponin are different in the fast and slow muscles. So meaning that, okay, then they might be that the calcium is affecting differently. But then in the EnvAd, there are some supportive proteins that are different in the fast and slow cells. And even in the jet disc, there are some supportive proteins that are different in the fast and slow muscles. So actually all the components of this contraction machine are different between the slow cells and the fast cells. But what about then the slow and fast muscles in the action? How fast is the fast cell? How slow is the slow cell? Well, these kind of comparisons are difficult to do nowadays because they were done already ages ago. So over here is one example from the 1960s made with a cat. And the cat soleus muscle, they're in the leg, and the extensor digitorum longus. They are quite often used as a fast and slow example of muscle cells or muscles. The soleus muscle is a little bit slower. You can see that the peak is a little bit later than in this EDL cell or EDL muscle. And then you can see that, okay, the tetanic tension is a little bit higher in this slow muscle. So there are some differences. But if, for example, in a single twitch, you must be quite carefully analyzing that, okay, is there any differences, etc. But in this study in 60s, they were comparing quite several different parameters. So the sarcomere length is completely the same. And this isometric tetanic tension is perhaps 10% higher in the soleus muscle. But the contraction, how fast the contraction happens, it's about 1/3 slower in the soleus muscle, so in this slow skeletal muscle. And the relaxation happens about 2 times slower in the slow skeletal muscle. But when they were comparing, actually the sarcomere shortening was happening faster in the soleus muscle than in this EDL muscle. And that means that, okay, if the sarcomere is actually shortening faster, there must be some other things than this sarcomere machine regulating that, okay, some cells are slow one and some cells are fast ones. But let's go them one by one. If you have different muscles, you have different neurons. And this is interesting thing that, okay, at least already in 1950s they were finding that, okay, they have different kind of motor neurons. They were actually identified by in 1950s by Ragnar Granit, a Finnish scientist who during the 40s already moved to Sweden to get the Nobel Prize, and finding out that, okay, there are some motor neurons that are producing stimulus much higher frequency, and some are producing stimulus a little bit lower frequency. So the firing rate in the soleus muscle is much lower, so maybe 10 or 20 times per second. But the duration of this, how long it will be stimulating, it can last for 200 seconds, so several minutes. But in this EDL muscle, 1 minute or even less, so even few seconds. And when they were collecting it, how many impulses per day there exist, in the soleus muscle you get 500,000 impulses per day, and you use it 8 hours, fully office day per day. So it means that, okay, you can then run these slow muscles for whole day, but these fast muscles only for short periods, 3 minutes per day or whatever. Okay, there are differences in the neurons. What about then on the neuromuscular junction? Okay, in this ideal neuromuscular junction, they are triggering higher local potential versus in the soleus muscle. So when you are giving the stimulus on the motor neuron to activate it, then with the same stimulus you get higher change in the postsynaptic membrane in the muscle cell. But these slow muscles, these neurotransmitters, are also metabolized slower. So they are there between the neuron cell and the muscle cell for a longer time. So meaning that because they are the release is on the acetylcholine, there is an enzyme acetylcholine esterase that is metabolizing it, so putting it in small pieces. And in these fast cells, in these fast muscle cells, they are metabolized much faster. So in the neuromuscular function, there's some differences. What about then on the EC coupling? Well, there are differences also between the slow cells and fast cells on the ion channels, especially on the sodium current. So if you have large sodium current in the cell, that makes that, okay, you have first local action potential. It will be spread all over the muscle cell quite fast with the sodium currents that are activated. But anyway, in these fast cells, the calcium is released 2 times faster. And their sarcoplasmic reticulum is also more developed. So that means that, okay, then you can have the signal from the neuron faster to a high calcium concentration. And that causes that, okay, now the tension is also produced faster. And there can be also some differences in what kind of channel you have there in the sarcoplasmic reticulum, because then you can allow faster the calcium release from the sarcoplasmic reticulum, and also there are some differences in the calcium pump. So then you're pumping back a little bit faster. Okay, then we have more calcium in these fast cells. What about then the contraction? Well, these fast muscle cells, they have now also far higher calcium capacity in the cytosol. So the free calcium is very low because it's binded in some other molecules in the cytosol. So that's why they have this capacity. But they will compensate it by having a huge calcium release. And also, there are differences in the sarcomer sensitivity for calcium. And this is interesting, that these slow muscles are contracted with lower calcium concentration. So you don't need so much calcium to release in these slow muscles. Well, you save energy because you don't have to release and pump back so much calcium. But the difference is then that, okay, when you are having these fast cells, they are producing so huge amount of calcium release, so it doesn't anymore matter that, okay, although they are less sensitive. But on the next video, we will discuss on more detail on what about then the energetic differences. Thank you.